Thank you so much. It's a pleasure to be able to speak to you. So I'm going to tell you about how I got started, and then how that even in a roundabout way led to helping develop the COVID vaccines and hopefully other messenger RNA therapies. If we could just have the very next slide. What I want to tell you is when I was a graduate student, if we could have the next slide, please. Yeah, when I was a graduate student in chemical engineering in 1974 and graduated, all my friends at MIT pretty much went to oil companies. They paid very high salaries and they had lots and lots of jobs. So I thought I'd do the same thing. But when I went to those companies, they'd say, well, gee, you could make billions, of profit for our companies by increasing the yield of one of these chemicals by 0.1%. And I wasn't very excited about that. And I started to think, how could I use my chemical engineering background to help people? I thought about teaching jobs, but nobody would hire me. So then I thought another way I could help people with my education was in medicine. So I wrote a lot of letters to people and nobody wrote me back. And then one day, one of the people at my lab said, Bob, he said, there's a surgeon named Judah Folkman. And he said, sometimes he hires unusual people. So I wrote him and he was nice enough to hire me. Now, he had an interesting idea, and that's shown on this slide, that really tumors need blood vessels to grow beyond a tiny size. Otherwise, they can't get nutrients. But he said that he thought that occurred because the tumors secrete a diffusible substance. He called it TAF, or tumor angiogenesis factor. And that enabled the blood vessels, which you see on the bottom, to start growing to the tumor. And then if you look at the upper right-hand side, you see as the tumor gets vascularized, of course, it gets bigger and bigger. And at the very top, it's very highly vascularized. And what happens then is the tumor can spread through those blood vessels. That's a process called metastasis. That is actually what often kills people. His idea, which was really revolutionary at the time and widely criticized, was that if you could, which I didn't know, but he said, well, if you could achieve anti-angiogenesis, that would be a whole possible new way to treat cancer. So my job, he said, it was to prove he was right and isolate the first substances that could do this. One of the key things in doing that, we felt, was to create a bioassay. There was no way to study blood vessels, so we had to invent one. The next slide shows what we had in mind. Maybe we could put tumors in an eye of a small animal, which had very little background blood vessels. And then we could maybe have a tiny delivery system that could deliver large molecules. All the molecules we were studying for preventing blood vessel growth were macromolecules like proteins or nucleic acids. And we needed that delivery system to be safe and to be able to be small and to be able to deliver any of these substances. But Dr. Folkman was on the board of the one company in the world at this time, this was 1974, that was working on drug delivery, a company called Alza. And he asked them, could they help? But if we could go to the next slide, what they all told us was that you couldn't do it. They said the molecules are just too big to diffuse through solid materials, just like none of you could walk through a brick wall. But if we were going to solve the blood vessel problem, we had to solve this problem. So I spent 2 years in the laboratory experimenting with different techniques, and I found over 200 different ways to get this to not work. But eventually, I was able to, if we go to the next slide, make little micro or nanoparticles. You see a picture of one and another cut in half. And we showed, and we published in the journal Nature, next slide, that you could use this approach for the very first time to release any large molecules, and also for the first time that you could release nucleic acids like DNA or RNA. I want to go back to 3 months before this paper came out in Nature. I got asked for the very first time in my life to give a talk at a major meeting. It was in Michigan. I practiced it for weeks, and I got up and I gave the talk. I was 27 years old, and I thought when I was done with the talk, I did okay because I didn't forget too much what I was going to say. And I thought all these older scientists, being nice people, would want to encourage me, this young guy. But when I stepped off the podium, a whole bunch of them came up to me and they said, we don't believe anything you just said. If we could go to the next slide, what they said was that the large molecules, if we could go to the next slide, please, the large molecules couldn't diffuse through the solid materials. And also the solvents we were using, like ethanol or methylene chloride, they would destroy whatever we put in. And things went downhill from there for me. Actually, I tried to get research.
It's behind him.
I tried to
The
get.
controller. It's coming.
No talking. MIT, please be quiet. Sorry, Bob, go ahead.
No, if we could go back a slide.
Yeah.
Yeah, basically they said that they basically said that that the large molecules couldn't get through the materials, and the solvents we used would destroy anything. And after that, my career really went downhill even further. My first 9 grants were rejected. I liked being a postdoc, but my friend said it's better to be a professor, so I applied for faculty positions. No chemical engineering department in the world would offer me a job. Finally, I got a job at the nutrition department, but Nevin Scrimshaw, he was an institute professor. He liked me. He offered me a job, but he was kind of a benevolent dictator kind of department head. He didn't ask anyone else what they thought. So a lot of them, well, he left the year after I joined. So a lot of the other professors told me I should go with him and leave. In fact, the next slide just shows what some of them said. Mike Marletto, a friend of mine at the time and still, was giving a speech on what it was like to be with me in 1979. Here's a quote from his speech. He said, one evening he went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So that was discouraging, but I kept working and on this, and I'm still here. And we were able to use these techniques to actually find the first blood vessel inhibitors. In fact, the next slide was just kind of gruesome, but there's no other way to show it. We used these systems in a paper we wrote in Science that same year. And what we were able to show is with 99 of the fractions we isolated, you get what you saw on the left. We literally did 2,000 rabbit eyes. Notice the sheet of blood vessels on your left. On the right, however, notice that the vessels are with the one inhibitory fraction we found. Notice that the vessels are lower, they're sparser, and they avoid the tumor. Now, this was a 1976 paper. Medicine is very, very slow. That's okay, you can go ahead. And medicine is very slow. And it took another 28 years from our paper, again, people using techniques that we developed, but another 28 years before the first blood vessel inhibitor would get approved by FDA. That drug is Avastin, which is one of the most widely used drugs in history. And there are many, many others that also got approved for all kinds of cancer, as well as really the very first drugs that could treat eye diseases in the back of the eye, like that cause blindness, like macular degeneration and diabetic retinopathy. So that was finally good to see. I want to go back to the 1970s, though. Dr. Folkman said to me one day, we should file a patent. And we did. And 5 years in a row, it got rejected. So the examiner said I should give up. And the examiner said this was obvious, and so forth. And people in the hospital said I should give up. But I don't like to give up. And I wondered, as I told you in the beginning, everybody told me this was impossible. It could never work. I wondered, maybe someone wrote that down. So I did a science citation search looking back at our 1976 paper. And I found actually articles that talked about what we did. If we could go to the next slide. And this, I had no idea it was written, but after 5 years of rejection, I thought this was maybe very helpful. These people said the agent to be released is a small molecule. One would not expect that macromolecules could be released because of extremely small permeation rates. However, Folkman and myself have reported some surprising. So I thought surprising, that's a great word for a patent examiner. That clearly demonstrates the opposite. So I showed this to everyone, and the patent examiner said, gee, he had no idea. He'll allow the patent if I could get written affidavits that these 5 people really wrote that. So I wrote them, and they were nice enough to write me back that they wrote it. And we got this very broad patent on the next slide. And with that patent, if we could go to the next slide, we would license it to different companies. I got involved in starting different companies, and there's all kinds of products affecting probably hundreds of millions of people that you see on this slide based on this. New ways of treating opioid addiction, like Vivitrol, ways of treating cancer, like Lupron Depot and Gliadel, ways of treating heart disease, like drug-eluting stents on the bottom right, and many, many others. Nonetheless, there's still problems. One of those problems is that when you make these things really tiny, they work, but sometimes the little particles, the nanoparticles, stick together Sometimes they're eaten by macrophages. So we had another idea. Next slide. What if we coat them? This was done with Vladimir Torchlin and others. What if we coat them with a FDA-approved molecule like polyethylene glycol, which is like a disguise? It'll have a lot of water. And the next video shows you from Nova, the TV show, shows you what we had in mind. Can you turn the sound on?
Sound up, please. Sound up.
Basically, what you were looking at was a nanoparticle with that special coating targeted to a cell and then blowing it up. Of course, it doesn't really blow it up. But that showed, but that type of technology, using polymers or lipids, led to what I'm talking about next. Next slide. That four of us, Derek Rossi, Nubar Fayan, Ken Chen, and myself, in 2010, thought maybe we could make a company by creating messenger RNA, putting it in nanoparticles to protect it. And the reason we thought this might be useful is that protein therapies by this time had become really revolutionary. Most of the top drugs by 2010 were proteins, but it takes years, a long time, over a year to make them. Very expensive. You need vats, or you need eggs, for example. In the case of flu, you have to guess what you're trying to stop, like H1N1 and so forth. Here, we thought we could do everything faster. You could make the messenger RNA quickly. Even from a computer, you could design it and then encapsulate it in a nanoparticle, inject it in the body, and then the body makes the protein. The body is the manufacturing plant. So that was the idea. The next slide just shows maybe we could do things that you'd never done before, that you couldn't just do normally, like diseases that were intracellular or membrane bound. So we started the company in 2010, and relatively quickly, I'm going to go through this fast, we entered in the next 9 years a number of clinical trials. If we could just go to the next slide. Okay, so that's right. So basically, we had over the next 9 years, we had made many different vaccines, and then COVID hit. And so we were in a position to actually hopefully stop this disease. And just this is the timeline. January 11th, you got the virus genetic sequence. January 13th, less than 2 days, Moderna scientists designed the vaccine, put it in nanoparticles, shipped it to NIH, and the first patients were treated, in March, just about 63 days later, which was after it started. And you do normally what's called a phase 1 trial when you do this first, which started on March 16th, to look at the highest safe dose. On May 19th, we actually announced the data. And I would have thought, the data was actually quite promising, I thought. But if you looked at the newspapers, the clinicians, the stock analysts, a lot of people just said, this is fake, it can't be right. Let me just show you on May 19th, the Boston Globe, our local newspaper, and their headline. Next slide. So this is their front page headline on May 19th, is "This is not how you do science," with my picture underneath it. So that was very discouraging. But let me just go 6 months later. After you do a phase 1 trial, you do a phase 2, then you do a phase 3. The phase 3 trial was 30,000 patients, double blind, so you don't know the results. And this 6 months later, in November, we broke the code. And I'm sitting in the boardroom on the board of Moderna too. And when the code was broken, the results were quite remarkable. Of 15,000 treated patients, not a single one went to the hospital. Not a single person died. And it was 94 to 95% more effective than the people in the control arm. A similar related vaccine made of messenger RNA in nanoparticles by Pfizer and BioNTech was also quite effective, about the same. And so this would get ultimately approved and used all over the world. The next slide just shows the Moderna vaccine. And I'll show you on the next slide some of the clinical data. And you can just look, at the enormous difference between hospitalizations, unvaccinated and vaccinated, and now vaccinated with booster. If you look at effectiveness, next slide, at the Mayo Clinic, The most effective is Moderna, followed by Pfizer BioNTech, also mRNA and nanoparticles, but a different type of nanoparticle and so forth. And then Johnson & Johnson, which is a adenoviral vaccine. The next slide shows a clinical trial, a large one, in Qatar, almost 400,000 people. And you can see the incredible effectiveness of mRNA, that's the Moderna one, even compared to the Pfizer one, in terms of infection. And the next slide shows over 400,000 patients. These are veterans, also in the New England Journal of Medicine this year. And again, you see the incredible effectiveness of these two vaccines. Again, the Moderna vaccine showed significantly less hospitalization than the Pfizer vaccine. Well, where do we go in the future? Well, I was visited, there's many different routes, and I'll just tell you one. About 10 years ago, I had a visitor who was a software expert, and he said to me, Bob, could you, I want to help people in Africa. Could you make vaccines that boost themselves? In other words, you could give one injection, and yet that injection will give a pulse at not only time 0, but maybe a month, 6 months, a year, so forth. If we could go to the next slide, I'll just show you a picture of my visit. That's me on the left and the software expert on the right, which is Bill Gates. The next slide is what he was suggesting. And if you look on the right, what you see are these pulses. That's what we really wanted to achieve. It had never been done before. And our idea in the bottom center is the way we would do this is we would put a nanoparticle or whatever, the red dot you see in the middle bottom, with the vaccine, and then cover it with a degradable FDA-approved polymer. And depending on the thickness of the gray and what you see, the micromolded particle and its chemical composition, it would break like an eggshell at different times. Let me just show you another video of how we make it. It's like a new printing approach.
Down, please. No, make that there. No.
Well, why don't we go to the next slide? Yeah, that's fine. So it's just a new printing press. It's almost Pico technology. These are just molds. The next slide just shows the first paper we wrote in Science, along with the patent we have with Bill. But the next slide is probably the most important thing. We've actually published now 5 science papers on this. Next slide.
Come on.
Next slide.
I'm trying. It's not working.
Well, why don't I just go over it? Yeah, basically, we basically got 11 pulses, and now we're even putting them into little Band-Aids that we can ship around the world. So anyhow, let me stop there, and I'm happy to try to answer any questions.
Okay. Thank you so much, Bob.
Oh, sure.
Thank you so much, Bob, for this incredible exposition of your life story and the successes. And also, I am so thrilled that you showed us all the negativity that you had to deal with. And I think that is truly important for the younger people around here. Never give up. Be more like Bob.
Thank you. Well,
So, yes. So, do people have questions? There might be some questions from the internet. Get ready. I'll give you the microphone. I have one question, and I know that Bob has to leave in about 10 minutes. So I have one question, though. Bob, a lot of people are queasy of needles. And I know it's not a big deal when we're comparing it to life and death situations, but is it possible to make the vaccine nasal, the mRNA vaccine? Can it be something that you don't have to go via needle?
By the way, that's one route. By the way, this is another route where you can't even see the needles. This is what I was going to show. These are microneedle patches. They're like little Band-Aids that Anna Jakulenik and Mark Prausnitz, who used to be in my lab, now a professor at Georgia Tech, are working on. And these, we're actually putting them right in these little Band-Aids, which could hopefully be shipped all over the world. So nasal vaccines are one, these patches are another, and so forth.
Yeah. Are there other questions? Yes, we have a question from the audience. Come and ask it from here. This is Dimitri, who is the co-founder and the treasurer of the Osmocosm Foundation, and also a lawyer.
Yeah, I have had dinner with your son, by the way, but not with you, Bob. But the question I have is, you kind of you referenced some differences between the Pfizer vaccine and the Moderna vaccine. Is there any difference in the manufacturing?
I'd
Or
say
what are the key differences between the two?
There are a number of differences. There may be differences in purity. There's definitely differences in the nanoparticles. There's differences in the amount, differences in the schedule. So there are quite a few differences. I mean, that's why I'm sure you see the differences in effectiveness.
Do we have any questions from the audience? Yes, Tristan, please come up here.
Yeah, hello, Bob. This is Tristan Roussel. Just a simple question. What does it take to take the decision to launch a new variant vaccine, like for the Omicron?
Well, I think the decision is
Yes.
really it's a decision that I think both Moderna and Pfizer and others make when there's such a need. You know, you decide to go ahead and you know, you could certainly do it because you have the sequences that you need to
Yes.
use to make the mRNA. But I think everybody felt that was such an important thing, and that we, you know, the team just decided to do it.
Great. Thank you.
This is Dimitri again, Bob. I have another question. I understand the fact that the Moderna vaccine is very helpful now. Is there any way that the technology can be utilized by Trojan horses or other viruses to kind of create the opposite effect?
Opposite effect in what way?
Like hurting people, actually creating, this kind of technology that is being developed can need be utilized in, ways that we can harm society.
Well, I would say, by the way, I mean, that's a fair question. I would say any technology, can harm society. There's no question. You could take this, you could take any technology, iPhones, different medicines, absolutely. I'm sure there are ways that people could do it.
And writing onto that question again a little bit more, Bob, we saw incredibly powerful evidence from you and data that this works to save lives, to save people from being hospitalized, to save them from having, a severe disease. And yet there's still people who will not take this vaccine, yet they will take other vaccines that are of an older technology. And I think that just giving them this data has not worked. Do you have a sense of why do we have recalcitrant people who are literally ready to bet their lives on an older and less effective technology for protection or no technology for protection versus when you show them something with this amount of effectiveness and it still doesn't convince them? I'm starting to think that the data alone is not going to convince anyone. What else do you think we could do to explain to people that this is a good thing to inject yourself with?
Yeah, I think it's a great question, and there's no simple answer. I mean, I would add, I think my understanding is over 40% of people in the United States don't think evolution is accurate. That there's no such thing as
40?
evolution. What's
No.
that? More?
Is it really 40? I'm so sorry to hear that. Is it really 40?
I think.
Please tell me you're 1-0 off. You have to be 1-0 off on this one.
Definitely not a 0 off. It may be higher. I was trying to be conservative.
Oh my God.
So, but I mean, the question's a really important one. There's so much misinformation, and people will believe different things for different reasons. I'm hardly any expert. I think some of the people in the Media Lab may be much better equipped to figure out why these things happen. But I do put it in a category like that. There's a giant number of people that do not believe in evolution. And I mean, just like what's correctly said, I don't know that data is clearly not enough to convince a lot of people of these things. So the question was very fair.
Hi, Dr. Langer. I had a quick question about earlier on in your career when you, even like recently, wherein the public really doubted your ability to execute on your projects and your visions. What really helped you stay towards your mission, stay towards achieving your vision that you had from the beginning, even when all these respected people would tell you that it's a fool's errand? So I just want to know more about your mindset at that time and what really helped you push through the negative outside pressure that you're facing.
I just believed in what we were doing. And I mean, I can't really say much more than that. I just felt it was, I just believed in it. And I wanted to try to do some good. And I didn't think that I could see some of the things like the delivery and the blood vessel things, which I showed you with my own eyes. So I didn't think the people that were criticizing me were right.
Got it. Thank you so much.
Sure.
So this new technology of the Band-Aids, I had a question around how do we make sure that they are equally dispersed among nations and people alike?
Well, that's a very good question. I mean, my feeling is that it's going to be governments that do that, in Africa and so forth. I think, our goal has been to work with the Gates Foundation from the beginning to try to create technologies that would be available for the entire world. In terms of distribution, I think that the foundation has people that hopefully can help on the various things they're doing to get them to the developing world. Because that's why we've been developing it way before COVID started.
Thank you.
Time for one more question after London.
Hi, Professor Langer. I just had one question. You did mention during your presentation that the technology that Pfizer and Moderna have are different. How do you think it affects people like myself that had the first 2 doses of the vaccine Pfizer, but had a booster of Moderna?
Well, first, I'm not a clinician, and I'm not sure anybody necessarily knows it. But people who do know more than me don't think it makes a difference. I mean, I think you could see the Moderna shots from the data are, somewhat more effective, but they're both very effective. And so I think that I think getting a booster, I think what you did sounds to me like overall a very good thing. I think it's people that don't get vaccinated at all, that are by far in the most trouble.
Okay, so this has been fantastic. Bob has to go to another engagement. Please, let's thank our speaker again. And thank you so much, Bob. This was amazing. Very inspirational.
Thank you.
Thank you.